Whenever there is a discovery of a new gene, the challenge is that what is the possible function of that gene? To find the possible function of that new gene, the most strategy used is to compare the sequence of the new gene with the sequences of the genes that are already known. And this strategy is known as sequence comparison. For example, one of the newly identified cancer-causing gene known as V6 oncogene, when the sequence of this V6 oncogene was compared with the available already sequenced genes, then it was a very astonishing discovery that the sequence of that newly identified oncogene was identical to a already known gene that was known as platelet derived growth factor. So that gene, platelet derived growth factor, is very important in the growth and development of the cell. So after this discovery, this was a very amazing phenomena that leads to the concept that most of the oncogenes, they are basically the normal functioning genes, but these normal functioning genes basically they are switched on and they code for a proteins at a wrong time. So it means that normal functioning genes performing their function or switched on at wrong time could cause to a cancer. So another example of that sequence comparison is that of the discovery of cystic fibrosis gene. So by using the same technique of sequence comparison, this gene was also discovered and found to be related to an already known gene. So in this cystic fibrosis disease that is basically found in children, and in America it is a very high ratio of this disease. So in this disease there is a large number of mucus that is present in the lining within the internal organs, and when this large number of mucus is secreted, so it causes, especially in the lungs, and it's caused the choking of the lungs, and it could be a very lethal in humans, especially in children. So in America there are 10 million people that are the carrier of these genes, and carrier means that they do not have the symptoms of the genes, but they have the tendency to cause the cystic fibrosis in the next generation. So when the two parents who are both positive and carrier to this gene, if they produce the offspring, so the chance that this disease will be transferred to the children is about 25%. So in 1989, it was pointed out that the gene of cystic fibrosis might be present on the chromosome number 7 in the human genome. And when it was pointed out, the location was narrowed down, and it was narrowed down up to a 1 million nucleotide. So still, after narrowing down the location of the gene, but still it could not be pointed out at a specific location. So the same strategy was applied. The strategy was sequence comparison of the sequences of 1 million nucleotides that was suspected that the cystic fibrosis gene was present within these nucleotides, and the sequence of these nucleotides was compared with the already known genes at that time. So the discovery was again astonishing, and the discovery was that the sequence of the cystic fibrosis gene was similar to the sequence of adenosine triphosphate binding proteins genes. So it means that the function of the cystic fibrosis gene was thought to be comparable to these genes, and these ATP binding proteins, they have a function within the cell, and they bound to the membrane within the cell, and they form the ion transport channel system. So we can now assume the function of the cystic fibrosis gene if it is related to that ATP binding proteins and they have function in the transport channel. So it means that cystic fibrosis gene also have some function related to the function of ATP binding proteins. So it was later assumed that cystic fibrosis has the function of, if it causes disease, it has some secretions, sweat secretions with abnormally high sodium contents. So we can say here that by using the technique of sequence comparison, we can find out the functions of the newly identified genes.